All right, great. Hello, everybody. My name is Kevin, and I am your instructor for. Very windy today, and I am your instructor for this course. Um, and I'm really excited to have all of you here. We're gonna today's lecture is not gonna be one that's like super duper rich, in my opinion, at least in like hard usable content. I did have some suggested readings. For those who aren't familiar, we did release a new syllabus. Although I'm working on finalizing that. And each week I will sort of finalize the suggested readings. For this week, not very much in the way of reading, but what I will say at the end of today's lecture again is that um, for those of you who don't have, oh my God, it's so windy. I'll just close this window. For those of you who don't have any chemistry experience, hello. It might be good to watch some YouTube videos on some basic chem or to flip through a chem textbook. And if you have any questions about that, we're always available to answer them. Welcome everyone. This course is going to be focused on a few things. It's going to be focused on taking the knowledge that you have, the fundamentals you have in biology. I know for some of you, you guys don't really have a lot of fundamentals. Maybe you haven't really had a lot of courses or things. And if that is the case, then you might have to work a little bit harder to read in advance and ask more questions. But of course, we will be here to support you. Otherwise, this course I think is pretty straightforward. Every Saturday, we're going to meet at this time to have a 2-hour lecture. The lecture is broken up into, I try to break them up with breaks, but often what you will find happens is we will get really deep into the material and we're gonna forget to have a break. I know last year people felt very strongly about having a break. So if I don't have a break and it's been about like an hour, you can like remind me in the chat and I'll try to find a stopping place. We have like a short break or something. I mean, I generally will need to have a break because my pet bird has to go to sleep, and so I have to cover him up. That'll be every night from 7 to 9 p.m. Eastern time, and then we will have the lecture's broken up, but it doesn't mean it's like two separate topics. It's usually just one topic. I sort of will sometimes it breaks up naturally, like we have 2 chapters of Campbell's or something. Note that Campbell's is our basis, but it's not all we'll cover. I had a lot of the material to cover in lecture goes beyond what's in Campbell. We might review a little bit of Campbell stuff in case people aren't familiar with it, or if I think it's really necessary to what we're going to talk about. And then every Tuesday there is a review. We called them office hours. I think that make people think that they were optional. They are optional, but like not really. You should probably go to them, where we will go over the homework. So each week we will assign homework and we will go over all of the answers. You will be graded, your grades will be tracked, and it will be told to your parents. Now, my grades don't mean anything. But you also probably shouldn't expect to get like an A in the class if your idea of an A is like 90 or above, because I don't think anybody who's taken this course has ever gotten that grade. Expect to score about a 50% if you're doing okay in the class, and close to like 70 to 80% if you're doing very, very well. And if you're doing significantly below that, like 10 or 20%, that might be something to be concerned about. But generally, depending on your level, and as you do more and pick up more knowledge, you'll do better and better on the assignments. For the first week, the assignment's very easy. It's a benchmark test. It's just like 10 multiple choice questions that we're going to release on Tuesday. Just so you can get an idea of where you stand and what you'll really get out of the course. And then every Friday, and this is the new concept / problem solving office hours on Friday. And this is a new element to the class. Last year, Friday office hours were more traditional, sort of like collegiate office hours, where we go over hints for the homework. But I find that isn't helpful. It just ends up being answers, but without us saying the answers, and then people still get the questions wrong. So we're just not going to have that. If you have trouble, just ask us directly what your question is, and we'll help you. A lot of students find that the homeworks are better to do in a group, so they form study groups. They formed organically last year. People want help finding them or whatever. We can help with that, and we will try to. We will obviously be able to help however we want. But what'll happen in the new concepts office hours is that Joseph will talk about either some things we couldn't talk about in a lecture, but more often than not, we talk about problem solving and how we tackle questions, and sort of some topics like, I'm restricted to a lecture because that's the format that we use for this. So I don't really get to put a ton of things on the board or to like draw for you guys. I will draw sometimes, not often, but sometimes. So that's going to be something that Joseph will do because he has a drawing pad and he's a fantastic artist. You'll get to see his work on Friday. For anyone who are curious, that is also include no payment necessary. So if you are still not sure about the course, you can obviously experience both of these office hours before having to make a payment. Also, if anyone ever has any comments or any questions, I have the chat open. I will read it. So feel free to say anything you want in there. All right. A few more logistics about the course. So each week, homework will be released on a Monday or Tuesday and then due the following Tuesday, obviously before Roy's office hours, because that's when we'll tell you the answers. And once we tell you the answers, right, there's not a huge point to you doing it. You can turn in homework late and there is a late penalty. It's just to encourage you to do it on time so your parent report looks better. I mean, like we grade all your homeworks by hand. So that's something that will happen. We'll figure out the submission process. It'll be on Canvas. All of you are currently in the demo class Canvas. We're going to have an official class Canvas for the students who actually decide to participate in the remainder of the session, and everything will be posted there. The readings are to be completed before the lecture. At least the ones given in the topics section. I'll go over what you should read each week before at the end of the lecture. The suggested readings you can read after. And I'm gonna refine them each week because I listed a bunch of chapters that I think are relevant and I think are really cool. And honestly, if you have the time, you should read all of the books cover to cover, except maybe Albert's, because Albert's is it's a very dense book and very, very long. But what I'll say is I will refine the chapters. Say, okay, look, I looked through them and I thought about it, and based on what we covered, chapters 2 and 4 of this book and chapter 5 of this book are the most useful. You can skip 6 and 7 or something, right? So you can read this after each lecture, but that's not strictly necessary. I think it's nice if you read these extra books, but you don't need to, especially to make semis. And people have in the past made camp top 20 with only reading Campbell's. We have a pretty bustling community, which I'll tell a little bit more about at the end of the lecture. And we're available by help via Discord, where Joseph will put the invite link in the chat. And usually I will stay after most classes if I don't have anything else going on, which, I mean, it's a Saturday night. What am I going to be doing? I'll stay after and help anybody who wants to be helped. Feel free to join the Discord. Some of you who are old students or took my other classes are already in it. You guys can talk about how cool it is. Thank you, Alex. How cool it is to join. We'll talk a little bit more about that again at the end. But I'm always on there. I'm always available by email. I'm a little slower on email. When I'm really busy, I only check email like once a week. So if I miss you, it might be like 10 days before I see your email. And then you can also I don't know, tell if your parents can tell Janet on WeChat and she can tell me and I will reply to her and she'll tell your parents and they'll tell you in the most roundabout way of asking a question known to mankind. You can do that if you want. Look, if you can find a way to reach me, I will answer your question. And then you will receive at least one progress report. We have tried to have more than one, but they are a lot of work to put together. And then all the students hate them. So it's kind of this lose-lose for everyone except the parents, and which of course, you know what they say, the money is always right. So we got to make these. So they will come out at least once. I'm just kidding. They'll probably come out twice, and then once at the end, just to assess your progress. I last year, to encourage people to do homework, I offered fun incentives. Like if enough people did the homework, we would make Joseph, I don't know, tap dance on video, and no one ever, they only ever met the thing once. So yeah, we will offer incentives again if people want them to do the homework, but you should really do the homework because they're good for you. But I know you guys are busy, and I know it's AP season, so I'll try to keep the first couple of homeworks light, to be mindful of that. If you can't make a lecture, you can let me know. We do tend to take attendance. That's only if you miss a lot. We'll tell your parents like your kid isn't showing up to class. But we do record everything, so you can always watch it in recordings. And yeah, we will. You can watch them, ask me questions after. They are usually posted in a timely manner. Usually. Sometimes it takes a while because Zoom is weird or like we have file processing issues. So anyhow, anyone have any questions so far about class logistics or anything? For those who are wondering about the homeworks are not like dumb. I promise you. I write questions that are usable style, short answer and multiple choice. The homeworks are all the same number of points each. Someone says I have to at 8. I don't know what that means, but thank you. If you can't stay for the whole lecture, that's obviously fine. You can watch the recording afterwards. But the homeworks are more similar to problem sets, where I give you a set of problems and you just do them, and they're for your own good. And if you don't do them, I mean, it hurts your grade, but I'm not like gutted if you don't do them. Actually, maybe I am a little bit. Anyhow, okay, it's time for instructor introductions. I realized that I forgot to make a slide for myself, so let me introduce me first, and then we'll go on to the TAs. So hello, I'm Kevin. I was a 3-time musical camper, silver medalist in my final year, bronze in my second. I've been teaching for many years, and I think teaching is one of the great joys in life. I've previously done a lot of private lessons, and with my private lesson students, I've had at least one finalist every year. We're still waiting on results from this year. I know some people thought the results were going to come out, yesterday, but they didn't. So we're going to be waiting on results to see how things are going. I have a pretty good working relationship with the CEE. I used to be a question writer. I don't write questions anymore because I teach, so it becomes of interest. But I still work with them on USABO related issues. So I'm really involved with USABO. I've been doing it a long time. I'm a Harvard alum, currently a genetics PhD student at UW Medicine. So very excited to be teaching all of you. But more importantly, you guys will be hearing a lot of me during this class. So I want to give a moment for my TAs to talk a little bit about themselves. Joseph.
All right, hello everyone. I'm Joseph. I'm a freshman in college right now at UPenn, Evangelist Scholar. Pretty sure no one knows what it is, but whatever.
Hold it right for the first and only time ever.
Yeah, pretty much. I am a one-time finalist, silver medalist. I feel like I could have got gold if I grinded a little harder, but
stop being salty.
Yeah, I'm just salty. I'm sorry. Yeah, I run the new concepts office hours. And yeah, semi-pro Brawl Stars, as it says on the slide.
All right. Well, Andrew, Joseph will 1v1 you, but if you lose, you have to sign up for this class, and the next one. All right. That's the policy. No, but Joseph is our go-to. He's like very good at chemistry and math, which are subjects that I am not quite as good at. I like solo bio. That's all I do. I only do biology, and everything I know about anything else is in the context of biology. As you can see here, here's a photo I stole from Joseph's Facebook of him eating pie in what appears to be an office storage closet. Oh yeah, plants. He is a plant main. So in bio, we joke about that people like have mains. Like you pick one topic and you get really good at that. It tends to be what happens because people gravitate to some subject. Joseph is a plant main, but he is not that good with plants. So I don't know. You can ask him plant questions, but the value is dubious. But yeah, so he's our plant main. I'll share more about you'll learn more about our history with biology, what we think about certain things as the course goes on. Lord knows you will hear too many of my opinions. So now let's give a moment to talk to our second TA, which is Roy. Roy?
why you decided to use that photo, but hey guys, I'm Roy. Right now I'm actually on a gap year and I'll be attending Harvard in the fall. So I do have a lot of other hobbies outside of bio, but I guess relevant to the course. I competed at IBO 2019 and I'm actually from Calgary, which is in Alberta, Canada. So if any of you have been to Banff or know where Vancouver is, uh, relatively close to there. And I run on mountain time. So anyone who's maybe from Colorado or something like that will be in the same time zone as me. But if I ever mess up my time zones and give you a time that might be hard to read from like Eastern time or Pacific time, just correct me. I might mess up there, but other than that, I'll do my best. Um, yeah, I think that's pretty much it. Looking forward to teaching you guys and enjoying the course.
Great, thank you, Roy, and hi, Claire. For those who are wondering, Roy is not a gamer. Whereas Joseph plays the game Brawl Stars, Roy is a real life Brawl Stars, and he will fight you and he will win. Last year, the homework incentive that people won was the workout office hours with Roy. And basically, it was Roy flexing on everyone how athletic he is for like 30 minutes, which is fine. That's what people wanted, so that's what they got. So those are our TAs. You can look forward to hearing their dulcet tones during their respective office hours each week. They're usually a good time. Roy's office hours are a little bit more participation focused, right? We ask the students to answer questions, and then we go over it like that. It's like being asked to go to the board to answer a question. What's 7 plus 8, right? Obviously 16. So everyone will be asked to participate. Well, not everyone, if you want to, and you can earn points, which you can redeem for terrible prizes in the server. Or otherwise, I don't know. Joseph is a music producer too, so he can make music for you. Oh, you're right, Sophia. It is 7 plus 8 is 17. It's obviously an odd number because 7 and 8 are both odd. So, this course has a few specific aims, right? We have a few goals in mind going into this course. First, we always want to make sure everyone gets into usable semifinals, right? And that's a very reasonable goal. Everyone who followed the course and for the most part and did the homework pretty much got into semifinals last year. And I think we could do it again this year, maybe with less chemistry. Our biggest complaint was too much chemistry. It's true. Everyone did technically make semis this year, so I guess I was very successful, but that was not my doing. All right, and obviously, hopefully, some of you make finals along the way. We have a few prospective finalists, and I'm really crossing my fingers out, hoping that they will make it. Results will come out soon, probably the next week, so we'll know then. But I think even more important than that, I want to make sure that everybody comes to have a strong background in biology. So if you want to do biological things, right, work in a lab, be a doctor, right? Pursue biotech, pursue bioconsulting. I want you to have a background for it. I don't want you to go into something, right, and then feel like, oh, I don't know as much as my peers. I promise that if you follow along, you will come out of this course and you will know more than most of your peers. Now, maybe one of your peers will be better at math, and that's just something you have to accept. But then you can console yourself in the fact that they will probably go to MIT, and then they will be very sad. I'm just kidding. MIT is a great school. And I also want you to have a passion for the biological world, right? I think, to tell a little bit about my story, when I was in middle school, the only thing to do, right, was competition math, AMCs, whatever. And I did it. And it was something that I did, and I was horrible at it. I am not good at competition math. I'm not even good at regular math. So I don't know why people thought I'd be good at competition math. And I would do I don't know if you guys ever did math counts, but there was that team round. I don't even want to begin. My strategy, right, for solving geometry problems was I would draw the thing really big and then I measure it and I'd be like, aha. And that was my strategy. So I was not good at math. And then, freshman year, of high school, my mom was like, "Oh, geez, this kid's hopeless." And in Asian mom language, she's like, "He's gonna work at McDonald's," because if you can't find the angle of A in a secant or whatever, you're never gonna have a job, right? Because that's how it works. Someone says I turned to bio because I couldn't qualify for USA JMO. I will tell you about. I never even got close. So then someone was like, "Oh, we're doing biology Olympiad," and I'm like, "Well, I'm good at knowing stuff," so I joined, and then oh my god, I fell in love with it. It was amazing because when you do math, right? I'm not walking around every day, with some guy who has 700 apples that he divides into prime segments that he's going to sell at a certain rate, right? That's not how my life works. If that wasn't how my life worked, I would leave. I would move somewhere else. But with biology, everything around me is something I could understand. Everything from just the fact that learning about biology opens your eyes to so many things. You look outside, you see a tree, and you can think about so many things. I remember I used to, I was kind of a what do you call those people? Spacemen. Space cadets. I was kind of a space cadet, so I would space out a lot because I would be thinking about things, thinking about how transpiration in the trees, I thinking about the cells in my body, the bacteria that lives on me, and it was just fantastic. And so I hoped all of you could come away with a little piece of that. I never even made Amy. The closest I got was one time I took the AMC 10 and I did I think 15 questions or something, and I was like, I feel okay about these, and I couldn't solve any of the last 10, and I'm like, you know what, if I guess on 3 random ones and I get 1 right, that boosts my chances by a lot. So I bubbled 3 randomly, and then I missed the cutoff by 1.5 points, and all 3 I guessed on were wrong. And so that's the story of why you don't guess randomly on a test where you get penalized. But on the usable, you don't get penalized for guessing, at least on the open, kind of, unless it's multiple true false, then you kind of do, but it's complicated, and we'll talk about that later. And so, and I want to lastly, and this is something that we found a way able to do later in the year last time, and I'm glad we're able to talk about it right now, is that we're able to hopefully provide a community of people who also care about biology and share the same interests as you, care about things beyond biology, to the people who are like alumni who might retake the course. Pranav and Claire are both whiz kids at coding, too smart for me. Kevin's brain's small. And I want to give you a community and a place where you guys can hang out. I'll be there to answer your questions about college, or why is your life so sad, or what's it like being an adult? And the answer to those in order is college is fun, life is bad, being an adult is worse. I'm just kidding. Being an adult is okay. You get to go to Home Depot whenever you want, and that's really the big. But I'm not that old, Roy. Home Depot is very good. You go to Home Depot, you don't buy anything. You watch the other people. It's great. So hopefully that's something we can provide for you, and I hope you have a good time in our course. What else is this course aiming to provide? For those of you who didn't see this ad campaign, we had 2 ad campaigns. We had one that was aimed towards parents, one that was aimed towards students. This was part of the student one. Our course is hopefully going to provide you with a fun experience. I am not that old. I want to make this very clear. I too am Gen Z, according to the to statistics. I have looked it up many times. So I'm here to provide a fun time for this, right? The class is these are some of the ads we had. I don't know. Please type 1 in the chat if you would have taken this, if you would have liked this course more if you known these existed. But anyhow, so hopefully this course you will find is a good fit for you. All right, now this part, I'm sure some people are like, why I'm talking about this, but I also want to talk a little bit about what is the exam. Some of you here may not know about it. Some of you here may know things, but not. Yeah, you'll find out. It's a joke later. You'll find out a little bit more about it. The USA Biology Olympiad is the premier biology competition for high schoolers, according to the CEE's website. It's got 3 rounds, right? The open exam, which hopefully you all will take. It's got the semifinal exam, which you can qualify for by being one of the top 500, and it's got the finals, which is the training camp where if you get top 20 on the semifinal exam, you get to attend a 2-week all expense paid trip this year to your bedroom where you can go to it on Zoom. On the next year, hopefully it will be back in person and it should be more fun. And you get trained, and then at the end of the training camp, you get the big payoff of getting to take a big exam the last 2 days. But it's kind of fun though, although it is stressful because it is still an exam. Joseph and I can tell you about it if you want. Finals camp is an experience, unless it's online, then it's. Yeah. But I will be lecturing for anyone who makes finals. I will be lecturing there. Someone asked, isn't finals top up 10%? Yes, it is, but that's only because top 10% is usually pretty close to 500, so they just take your top 10%. In the past, when it was 10,000 people, it's actually shrunk over the years a little bit. It used to be 500, and now it's still about 500. So, if anyone who's wondering, that is about the cutoff. It is top 10%. So here are the 2021 open exam results. They're pretty normal, I think, for what you might expect from a year. So it's like slightly fewer people than normal. But if you scored a 1, like, congrats, you did worse than guessing. That's impressive. I feel like that must be former IBO competitors smurfing. And then nobody in my class scored a 43, but I do think we have one student score a 37 was our highest score, which is very respectable, higher than anything I think I ever scored, to be honest. The open exam scores are a little random, because they're very, very prone to what we call usable moments, where, or usabra moments is the name we gave them towards the end of our final class, because they have a lot of mistakes sometimes, and you never know until they get released 2 years later. So sucks to suck. But yeah, the 2 exams are a little bit different from each other, and they have really different test-taking strategies. During this course, because everyone takes the open exam, a lot of our stuff is going to be shifted towards open level practice. And you're thinking, but Kevin, I'm already very strong. I don't need open level practice. Yes, you do. Everyone needs open level practice, because open level practice is the same thing as semifinal level practice, because they're basically the same exam, just one's longer. The questions don't actually differ in difficulty by that much anymore, especially on part A of the semifinal. Semifinal is 2 parts. It's part A, 50 questions, part B, 50 questions, part C, free answer. Part A is pretty similar level now to an open exam. And then part B is significantly harder, but part A is very easy. So it's not, so by preparing for this, I want to train everyone. I think one of the saddest things I see in students is so many students are so ambitious. They've been studying biology for 1 year, and they're like, I'm going to read Albert's Molecular Biology of the Cell. And then, they get a question that's really easy, but they forget the basics and they get it wrong. And I don't want people to suffer from that. So we're going to focus on reinforce the fundamentals while also building on top of that, right? A lot of people study on their own, and I've had a lot of students in the past, right, who have this problem where they know a lot of stuff. They can tell me exactly, like, the pathway for pyroryanidine receptor synthesis, but they cannot tell me the anything about calmodulin, and that's terrible. This makes no sense to you now, but hopefully at the end of the course it will. Someone asked, so semifinals are only multiple choice? No, there are 1 or 20 multiple choice questions and a short answer portion. They've had essay style ones in the past, but they don't really do those anymore. The multiple choice questions make up the bulk of the points, but there's a significant number from the short answer as well. Both are prone to having hilarious errors. No diagram, no question, no choices. They've all happened. Life can be tough, and apparently the people who do the quality control at CEE also find it tough. I'm also going to talk about a few people, because they're people who I know, and I'll reference them sometimes. And I know people were like, "Who's this? Who's that?" Yeah, one time they had a picture. There was no question, and they had choices. And we were like, "What was the question? What are you asking?" It was just like a graph, and no one knew what to pick. To this day, no one knows what the question is. Okay. Consider the following: no picture and just choices. Here are some of the big wigs at USABO. This is Kathy Frame. She's now like special consultant. She was, I think so, Pranav. She was the director of USABO back in my day. But now she's special consultant. She's great. We call her Mrs. Frame Perfect because she's must be a speed runner because with how fast she must check the USABO exams, she's clearly not finding any mistakes. So this is Mrs. Frame. She's great. I know her. She and I text sometimes, like not like, hey girl, what's up, but just like, hi Mrs. Frame, how are you doing? I couldn't find a picture of Michelle King, so this is a stock photo of a woman named Michelle, and I put a crown on her, so she's a king. Michelle King is the current director of USABO. She's very nice. I actually met her. I just couldn't find a picture of her. So she's the current director. She's very nice. I don't know that much about her. If you go to finals, you will meet her. And then this is Joanne P. DeJaneiro. She is the director of the CEE. Rumor has it, and the joke around the block is that she hates USABO. And I don't want to say that as if it's like fact, but like she does not appear to care as much about USABO as she does about RSI or just the other program the CE runs. And I guess energy is divided. Sometimes I will make the joke that it's not delivery, it's DeGennaro. And that's a reference to one time I was at a CE thing and I accidentally called her Miss DiGiorno and they got mad at me. Well, there was a comment on the Facebook post actually. It was an event, and then they replied with, "It's not DiGiorno, it's DiGiorno." And I'm like, "Oh, sorry, I thought it was delivery." And they didn't seem to get the joke, and I got blacklisted from their Facebook for like a year, but that's fine. She's a very wonderful woman and has a lot of history in education. That's all I will say about her. So. Last year, at this point, we were to begin talking about like Campbell book stuff, but I figured that that's not quite as important. Let's talk a little bit about key strategies for the exam. And these are things that I hope I can teach you guys by the end of the course, right? They're not things that you need to have right now. If you have all of them right now, well, maybe you don't need the course. But I think it's pretty hard, and you will find if you take some practice tests that it's hard to have all these skills, and we're here to teach them to you, right? So the first is reading and interpreting. Now you're thinking, Kevin, I know how to read. I've been reading since I was in kindergarten. I was reading at a 12th grade level when I was in 7th grade, and sure, but USABO requires you to read at a 7th grade level in 12th grade because apparently nobody can read the words not, except, all but, are all words that people don't seem to see. And they get the question wrong, and they're like, what? How? And I have to explain, and it's very embarrassing for people who ask. So we're going to talk about reading, but also interpreting the question. Sometimes questions are worded in ways that give away more information than you might expect. And that's something that's important. Also, because the questions today are so long, you have to prioritize what you're reading and what order you read things in. And that's another thing that we're going to try to help with. That, segues into our second point, which is speed and pacing. The USABO exam is pretty tight on time. I remember when I was younger, when I still did math, they were like, oh, AMC 8 is tight on time. AMC 10 and 12 is tight on difficulty. USABO says, no, fam, why don't we be difficult and tough on time? And so it's tough. It's a really hard exam. The semifinals is legitimately tough. A lot of the questions are like really long, take a lot of brain power, and they're mega long. They're like a page long each. So you have to like read a lot. So you have to know how to pace yourself, right? For the open exam, it's 1 minute a question. For the semifinal exam, you have to, on part A, pretty much do 2 questions a minute. And you don't really have a choice because part B is really long. It's like mega long. Like, and then part C, you have to write the answers down. So that also takes time. I will say that the big thing is I will this year for training on part C, I'm going to encourage people to write short answers. Like I read so many people who wrote me like I graded some of the USABO semifinal. And some people would write me these essays about why the mode of inheritance was autosomal dominant. But it doesn't matter if you're wrong. It doesn't matter. You're still wrong. So all you have to do is like write one explanation for why the correct answer is right. So write quick, short answers that are concise, and that will get you the points. Biological intuition. Not always will you know everything. A lot of usable questions today are based off papers. Luckily, a lot of them are written at least pretty decently, so you can figure out the information. But there's really no. People think that the strategy for usable is memorization, and yes, you have to know stuff, but that's not the way. If you try to memorize everything, you will fail because there is so much information. What a throwback. Someone talking about MySpace. Instead, you need to have intuition, right? The things they're asking you about are things that are gonna relate to things you already know. And if you can draw those connections, you can infer new information and then answer the question without necessarily knowing exactly what they are talking about. And then lastly, this is something I borrowed from video gaming because Joseph and I are big gamers. I just kidding. I don't game that much, at least. But to be tilted means to sort of get bothered. I think a lot of people, they'll like get a question wrong, or they'll like know they know the answer to a question, and that's a feeling that everybody's had. Everyone hates, right? When you look at a question, you're like, oh my god, I like definitely knew this, and you just can't remember. That can get you all bothered, and that's going to affect us in your test. And we're going to talk about how to not have that happen, in part by not having that happen, where you know things that you know, everything you think you will know, and how to properly figure out what is the best way to get yourself to an answer that you can be pretty happy with statistically, and then move on. So those are some of the skills that we hope to teach you. The other thing we will talk about is this idea of the USABO meta. The meta is this idea that in the game you have to play the game of the game, right? It's like how I'm trying to think of something that everyone like basketball. If you're I don't know how basketball works, so you guys can tell me if my analogy is very wrong. But imagine if the other team had only shooty people, like people who throw the ball. Then maybe your strategy is to get really, really tall people to act like a wall and block them. Now, you might not score as many points, but if the opponent can't throw the ball over the really tall people, then you're going to be able to win. Is that how basketball works? No? All right. Well, basically, the idea is the usable is something that you can also sort of take advantage of, right? You don't necessarily have to know all of biology. You just have to know what they're going to test on. And by knowing that, we can sort of get ahead of the edge, right? There's some information that used to be really big, like critical to know 4 or 5 years ago. I think the best example of this is probably, this is like memorizing immunoglobulins was like huge a while back. Like it was, I think like one of the big things you had to know. It was like the go-to know topic for when I was competing. Today, knowing it's like useful, but it's honestly not that good because they don't test on it anymore, right? So that's kind of how it works. And so hopefully we have a lot of experience. We looked at a lot of tests. God knows Joseph and I spent a lot of time looking through these tests. So we should be able to help you figure out, what to do and how to maximize the amount of effort you put in. All right. So now we can move on to material. Before we do, any questions from anyone? You can ask in chat. You can unmute and talk if you want. Anyone have any questions? I know people probably feel a little shy, but I'm very friendly. So Bacon? You guys know what Bacon? This is a reference to Francis Bacon, the father of modern science. Yeah. Anyone else have any other questions? Especially you guys about usable or anything, I'm happy to answer them. And the other question, we'll move on. I wish we could talk about bacon, and we won't get to. There's no lectures where bacon's gonna be like a real topic, unfortunately. Oh my goodness, we're not talking about lasagna anymore. So let's talk about the scientific method, right? So everyone here has been in 7th grade science. The teacher was like, "Okay, everyone, let's make an observation." And everyone like makes an observation and they're like, wow, now we know that the light bulb turns on when you press the switch. But that's not how it works. If you want to get the result published, you better get some significance. So you better test many, many lights over the course of many switches to determine. There is, darn, I'm sorry to hear that, Sophia. Maybe you never got to the light switch part. So this Campbell's has this example. I don't think it's in 10th edition. This was like back in 7, where they were like, what if you have a flashlight that doesn't work? And the idea is, what is science? So science is something that you can test. There's this big debate as, can things that are presently untestable be science? And the really conservative answer is no, which is why dark matter, like 40 years, more than 40, like 80 years ago, was not science. It was hoopla. Like no one could test for it. It's pretty theoretical. So it was like kind of science, but more so just like theory, not even like real theory, because real theory is supported by evidence, right? But so only things that are testable can be science, right? So if your flashlight is broken, blah blah, the idea is, you know, let's say, you know, you're with 3 friends, right? Me, Joseph, and Roy, right? And then, you know, we have a broken flashlight, and I said, okay, what if the flashlight is broken because it's haunted? The last person to use this flashlight, you know, died tragically, and now they prevent that from turning on, right? And then Joseph, being very reasonable, says probably it needs more batteries. And then Roy, being creative, says probably the bulb is burnt out. What I suggested is not testable. There is no current scientific instrument to test for presence of ghosts, right? Maybe one day there will be, and then this will be science, but today it isn't. And so the other two have hypotheses that can be tested, so thus there's science. And we can design experiments and collect data, and then I ideally publish them. You know, I'm a scientist now, so whatever I do, I try to think about how to publish it. So yeah. Now, science fundamentally works from the direction of deduction, right? Where you begin with a theory, right? Your theory, for example, will be animals change over time, and they have a hypothesis. And we'll talk a little bit more about that in a second. But you have something that they change because, you know, in Darwin's case, he was like, because there's too many individuals and only the most fit survive. And then he goes and looks at big turtles, and then after looking at enough turtles, he's like, yes, my theory is correct. He actually looked at more pigeons than turtles, but we'll talk about that when we get to the evolution lecture. So, and then induction's the opposite. Now, can we not do science from induction? That is not true. There are cases in which induction can help us reach theories, right? Notice how this can go right here. A lot of philosophy is based off induction. And so it's important that we have both. And that's why you guys should study humanities. This is the big argument for why people should study humanities. It's not a great one, there's better examples, but this is just one. They have never tested this on Usable. I don't think it ever will be tested, but they could. So I guess, whatever. All right, let's talk a little bit about experimental design. All right, interesting. Yeah, kind of dark in the chat. So Usable today has a lot of experiments. Basically, most of the exam today is I look at an experiment and I'm like, okay, what conclusion can you draw? What is good about the experiment, etc., etc. Sometimes it's used to test methods, sometimes it's used to test your ability to draw conclusions, but sometimes they directly test your ability to look at experimental design. And in these cases, what happens is you have to first think about why are they doing this, right? You have an experiment where, we have a bunch of cells and we expose them to tunicamycin, which is a protein folding inhibitor. You have to think about why they're doing it. Luckily for us, Usable people, most of them will never like produce original data and then like slap it on the Usable for you to look at, because first of all, that's bad. You should never do that. But next, like papers that get published, they're usually okay. Like their methods are usually sound. That's not always true. There's some bad papers, but the paper ones that you read are usually gonna be okay, because the bad ones, no one looks at them, so they get sort of self-selected against. You gotta think, why would an experimenter do this, right? And maybe they put in tunicamycin. I'm gonna assume they're probably trying to test for whether or not a protein is expressed in the presence of misfolded proteins, or whether or not this other thing can inhibit it. So we've got to think about what they're testing for. And then you also think about the hypothesis, if any, is given to you, right? And hypotheses are, you want to rephrase them in the statement, if X, then Y, because Z, right? If the flashlight is broken, because of a bad bulb, then replacing the bulb will fix it because the bulb was the broken element, right? But it can get complicated, right? There could, and the things go against hypothesis. I think what is wrong is X wrong, Y wrong, or Z wrong. And usually it's like Y or Z, and sometimes it's X. But like you tested the wrong thing and didn't give you what you expected as a result. So there's a variety of ways that things can go wrong. And to do this, most experiments need both a positive and a negative control. So in the case of the flashlight, we don't have a genuine positive control, right? And think about the flashlight experiment earlier. The issue may be neither the batteries nor the bulb. It could be an internal wiring default of the flashlight. And so what you do is you need to have a working flashlight, take the batteries from the unworking one, put them into the working flashlight, and then take the working battery, put it in the unworking, and then show that they switch results as a positive and a negative control. If you just replace the batteries on the flashlight, you don't know if that actually fixed it. Maybe it wasn't that the batteries were dead. Maybe one of the batteries had like a little, like some sort of like sticky substance on it that prevented it from conducting electricity, and that's what caused it. The batteries weren't dead. You can't prove your hypothesis unless you test against both the positive and the negative. If your theory is the battery is dead, you have to first show that a live battery would restore the power. You have to show that the current batteries don't have any power. So you need both positive and negative controls. Consequently, as a result, we are, I guess, in many ways, like, you had to test for more things than you expect. That's why if you like ever, one of the big pieces of things that I used about today are electrophoresis results, and because Western plots are so amazing, it's just easy to interpret. And you're gonna see that a lot of them just have columns where you're like, this experiment doesn't seem to do anything, right? Like for example, they want to test if knocking out gene A changes the expression of gene B, and they'll do an experiment where they like add in a second copy of gene A or knock out a gene that's unrelated, and you're thinking, why are they doing that? That's a control. It's showing that the act of knocking on a random gene isn't affecting the thing, right? Like, for example, if your hypothesis is, if I light my mom on fire, then she will be mad because people don't like to be on fire. You have to light your mom, but you also have to prove that it's not another thing. Maybe she's not mad that she's on fire, but rather that you were playing with matches, right? So you have to play with matches and then not light anyone on fire. And then you also have to show that if someone else lights her on fire, it would also make her big. What if it's you lighting her? So you got to test for a lot of things. So there's different examples of different ways you could test for these kinds of things. It can often be really difficult to get positive controls. For example, if your experiment is gene X causes cancer, what are you going to do? Put gene X into a person? Like, you can't do that. And so there's like limits to what you can do. In some cases, usually how they do this, they will look for individuals with like a mutant gene X or whatever, and see if they have cancer. And there's one, that's why medical science can be kind of slow, because they do have to wait until some of these things show up. And that's why rare diseases are almost impossible to study, because you have like 6 people have a disease. They may all have different genotypes. There's just really no way to tell what's causing it sometimes. So anyhow, positive negative control is something really important to think about, and one of the things that does show up on the test, and it can really throw you off if you're trying to find a reason for things. But the reason could be, oh, it's a random control. And this is one of those things that you will build. And I'm thinking, is there a definitive way to tell? And I think yes, to me there is, but it's hard if you don't have biological intuition. So by doing more tests, by looking at more questions, you can build up that experience that's going to make you better at doing these kinds of questions. Oh, and I put this here again in case anybody want to ask about this. What would an example of a positive or negative control be here, right? Like, for example, in the batteries example, you would take dead batteries, known dead batteries, or rocks in the shape and design of batteries, and put them into the battery to prove that the battery doesn't turn on. Imagine if you remove the batteries and you put in rocks into the batteries and it turned on, right? That would like throw your whole hypothesis off. And then if you put in like working ones and it still works, then there's a lot weirder things going on. Then you might have to go to Kevin's "it's haunted" hypothesis, right? So I'm just kidding. You would never go to that because you can't test it. Albeit, there have been things that we've discovered that we couldn't test. There was a really interesting article written in Physics Today, where somebody did an experiment and they showed that if you freeze water first, and then you take it from frozen and you try to like at 0 and try to boil it. It boils faster than water. Like it takes water, you freeze it, you let it melt to room temperature, and then you boil it. It boils faster. Wait, no. If you boil water first and then you freeze it, it freezes faster than if you just took the water and you try to freeze it. And that like defies all logic, right? It should be the same. And there was a really interesting thing about figuring that out, and like people were like, it's. Some people were like, it's God, and it's like no. They have hypotheses for it. They're actually testing it. There's some really cool things about it. So yeah. All right. I see that we're bordering the hour mark. We have a pretty big section coming up. Do people want a break, or do people want the last half of this lecture is pretty light? Do people want a break? Do we, people say something? I can't really see reactions right now because I'm not in like the normal Zoom mode. What do people think? Break? No break? Okay. Why don't we give everybody a quick 5-minute break, and then we will resume. How about that? All right. So right now it's showing on my clock 6:56, 7:56 for East timer. So we'll come back at 6:51. So everybody go, get water, whatever you need. How's everybody doing? Everybody good? Well, we have time for a break. It's lecture 1. It's not super duper intense. Well, I guess if you guys don't want breaks, that's fine. I don't need to have them. I mean, sometimes I need them because sometimes lectures are really intense. But yeah. So I usually get Campbell's from, someone asked, where do you get Campbell's from? I got all of mine from Amazon. There are some other websites. Any used copy will do. There's also some places to get there are also some places to get like PDFs that are questionably legal. I'm not going to talk about it in case I get clapped, but yeah, you can get them in a variety of places. So, but I recommend Amazon. You can, if you're looking for like a really cheap copy, 7 will do. 7's really cheap now. It's missing some information. The chapters might be a little outdated for some of them, like the genomics one. But I will say Campbell's genomics chapter is pretty bad. So it's not like you're going to miss a lot by having a slightly less updated version. Do I like the 7th or the 11th edition more? I've never read the 11th edition. I've currently only used 10, which I actually, so I have a, so you guys will see during class, I look this way, I look here because I have a monitor here, and then I have a music stand over here that I put this copy of Campbell's on, so I can always look at it. I actually think this is a great investment. I actually really liked this back in the day too, because I was a musician back then. I played flute. To have a music stand and then putting your books on it is a great way to like read without having to like hold a heavy book or sit on a desk. Because my desk was kind of low, I'm kind of tall, so it was nice to have a music stand. So I like the 10th edition. I think it's okay. I think 9 is not good. 9 is very, very no, 8 is not good. 8 is like really bad. 9 is okay. But I'd say 7 and 10 are comparable to each other. Yeah, I mean, not on music. Like the wire ones don't work. You gotta get one of the like the solid metal ones. Edition 12 would be fine for anyone who's interested in the 12th edition. It's hard to get a copy of it. It's kind of expensive. 10 is okay. 11 is okay. Yeah. So I also have a PDF of 12. I just, I don't think 12 is going to be radically different from 10. Campbell's is a very conservative like book, and that's one weakness of it. I will say is that they don't update. Like they don't put new information in. They're more likely to take information out than to put information in, which I really don't like. So the big difference between 10 and 7 is that in 7 you don't have Illumina sequencing. That's the only thing I will say that's like a huge difference. But Campbell's doesn't explain Illumina sequencing like super duper well. It's better than most of the online stuff I've seen. If you don't have a good background, but yeah. Someone asks, is it really that important in USPO? I assume they mean is Illumina sequencing? I think more so. Although they still test more on Singer. So I think that's gonna be like something that you definitely need to know. But knowing Illumina probably helps. I haven't seen a ton of like next gen sequencing questions, but I think they're gonna start creeping in, if not outright taking over Singer questions, especially on open, where it's more recall based. So, oh my God, we're supposed to start again. Is everyone back? Well, people aren't back. Won't know. I guess we'll give them a minute or two. But how are people liking the lecture so far? Everything good? People have any comments? Do you want me to go faster, go slower? Some people say I talk really fast. So people don't always love that. Great. I'm glad that people are enjoying. If you don't like, you can DM me. You don't have to publicly be like, your lecture's terrible. I've never seen a worse lecture in my life. You don't have to do that. Unless you want to, I guess. I'm fine with that too. Okay. I'm gonna assume that people have had a chance to use the bathroom, get water, eat dinner. I know this is it's hard to have a time that works for everyone because we are like a nationwide sort of experience. So anyhow, I do talk kind of fast. Yeah, it's kind of a problem. So especially when I get excited, I talk really fast. So if I ever start doing that, you can just tell me, and I will try to slow down. But then I'll probably speed up again. Okay. Anyways, data and methods. The heart of science, anyone who wants to do science, is data. All right. All the time, my professor's like, Kevin, where's the data? Why don't you have data yet? It's been 3 weeks. Where's the data? And then I have to be like, well, you see, the experiment didn't work. And that happens a lot. It's truly tragic. You will do an experiment and you'll spend like weeks on it. And you'll be like, this is gonna be perfect. And then you put the thing into the machine or you incubate it. You put and then it comes out and it doesn't work. It's like baking a pizza. You put the pizza in and you're like, I'm gonna get it, wow, delicioso, and then it comes out, you're like, what is this? This is disgusting. And then you're like, oh, I didn't put any crust, and that's why your thing did not work. It happens. And that happened to me 2 weeks ago. I put a thing in the machine. I'm like, this is gonna be great. And then it came out. I'm like, what? Why is all my data 0? And I forgot to put any cells in the cell counter. So obviously it came out to be 0 because I'm stupid. And that was 2 days wasted. And now I'm off tempo and I have to restart. So everything sucks. But data, at the end of it, what we produce is figures. Now, for anyone here who's interested in reading papers or doing research, you're gonna have to read a long journal papers, and they suck because they're gonna be to do the experiment, we put 0.3 milliliters of this into the machine using the and that's terrible. So everyone skips, as far as I know, every professor's, postdocs, lab techs, They all skip the methods and data part of the journal because it's useless. What are you gonna do? You care about the conclusion, you care about the figures. The figures, from that you can figure out what they basically did. You don't need to know like specifically, we added this reagent. I do this because pipetting. We added this reagent and that caused this. You don't need to know that. What you need to know generally is, oh, here's what they were doing. Here's the idea of what they were trying to do. And that's why they got the result that they wanted, right? Going back to our experimental design section. Now, some people will just like dump all their data on you. They'll be like, we collected 3 million data points. You can find it in this Excel spreadsheet. And they like have a link to Excel spreadsheet, and you should never click on it because it is a trap. You will open that Excel. Your computer will not work again for the next week. You're just trying to open the stupid Excel file. So what we got to do, we're going to take the data and we have to collate it into a way that can be looked at. So we have to visualize it. And generally speaking, experiments have data in either one or two forms. Binary data, so presence, absence, red versus green, has 4 legs, does not have 4 legs, right? Binary data, or they have it in float data, which is like numbers. Usable for the most part, you're gonna be working with binary or like somewhat of like a subjective gradient of data, because no one expects you to do math or statistics except maybe on a part C. So you're not gonna be working with statistical data. So it's important to learn how to interpret these binary data points and what they mean. I should say discrete versus continuous data, to be more specific. So let's talk about data visualization, right? So the big data tables are really bad. So you gotta make them work for you. But the issue is most figures are made in like 4 programs. And all 4 programs have limitations. Figma is kind of the Porsche, right? It's the best one, but it's not everyone uses it. Office is like by far the worst, but thank you, Alex. I was thinking that. Office is by far the worst, but it's by far the easiest to use. R is like in the middle, and then Photoshop. Photoshop is for our old boys, the people who never figured out how computers work, and then they're like, so if people who use Photoshop, they will like use the pixels, and they're always correct, but it's very, very like it's a very, very time-consuming process. So people use Photoshop to make figures like they're cut on another, cut from a different cloth. If Joseph ever became a scientist, I'm 90% sure that he would make all of his figures in Scratch. He has a long history as a Scratch programmer, so for anyone who's interested in that, you can bother Joseph about it. So now let's talk about different types of graphs. There are fundamentally only 3 types of graphs, and really there is only 2 types of graph. But one of them is different from the other one. So we can begin with box and whisker plots. Box and whisker plots basically plots all of your data points in a way where you can easily see how they're distributed. Now, thinking, Kevin, this plot is very simple. Can't imagine a real scientist ever using this. Boom, you're wrong. These are both from Nature papers. Well, this one's not. But these are figures from a Nature paper, a paper in Nature. This paper was called distribution of things expressed as box and whisker plots in different types of sediment. That's how I found the paper. So this is a very common data type. And you're thinking, why? Well, it's because it shows you the one thing scientists really care about, what the average is, and then how big the spread is. Note that in the box and whisker plot, these lines are not error bars. These lines these lines are either range, but more often they are 2 times interquartile range. So that means that they're not actually showing the max range because you have outliers. They've excluded them and they're actually showing like 2 times the interquartile space, or like double the amount of stuff in this section. So these box and whisker plots are kind of deceptive because they don't tell you really like how concentrated things are. These are violin plots. They're like box and whisker plots, but they show you like how the data is actually distributed. They're called violin plots because these look very vaguely like violins if you have never seen a violin before. And these are very, very common as well in data, and you'll see them sometimes. These actually, we're going to see, bleed into other data in that they are actually really weird histograms. So we'll see that. The next category are histograms and bar graphs. Bar graphs are pretty, you've all seen it. these depict frequency over a range, right? So while Box and Whisker is limited in that they only show distribution of a single variable, these show distribution over a range. And this is actually, I actually took a bunch of these figures from the 2020 semis. Yeah, I think maybe they do resemble cellos a little more. But this is specifically flow cytometry data. And one of the critical skills that people seem to not know is looking at axes. One thing you can see with these is you got to figure out what's on each axis. So immediately by looking at the x-axis, I can see that it's some sort of logarithmic distributed scale, right? And then we know that each of these has to then be some sort of like either intensity or something. There's not very many log distributed variables that we use commonly. And then the y-axis is always going to be frequency, right? Being able to identify quickly that this is some sort of histogram gives you a lot of power to immediately know what you're looking at. So you're looking at, okay, there's more stuff here and there's a narrow peak here. So it's an informative graph. Lastly, you have line graphs and slash plots. These are fundamentally the same thing as scatter plots, where you're showing how two variables relate to each other. In this case, we're showing current versus voltage. Again, 2020 semis. We may practice this exam at some point if during one of the booster classes that sometimes we offer. We'll have to see if we offer those again this year. But they show a relationship of two different variables. Okay, so we're going to play a game. Everyone get ready on your keyboards to type in the answer. Of the three types of plots we've shown before, what type of plot is this? The choices are box and whisker, histogram, or this is an easy question for now. Don't stop complaining. Box and whisker or line graph. Which one is it? Everybody put your answers in the chat. All right, I got one answer. Anyone else? You can DM it to me, say it publicly. All right. A lot of people are saying histogram. If you said histogram, you are wrong. This is, in fact, not a histogram. It is a line graph because it is showing two different variables in relation to each other, and it is not, in fact, showing frequency. Right. So this is actually a line graph, right? Even though they filled one of the lines in, that doesn't mean anything. They just filled it in. You see? And that's how you get big brained. And that's why you have to know what kind of graph. If you're thinking this is frequency, it looks similar to this one, but it's not the same data. This is plotting frequency data. This is plotting two different variables against each other. All right, we got one more. Okay. Tell me what kind of plot is this? This is not from the semis, Pranav. It's fine. This is from Kotian et al., Nature Genetics. All right, I've seen a couple answers. Anyone else want to try? Okay, people are generally saying box and whiskers had a plot. You are also wrong. This is, in fact, also a line graph because this is plotting location on the chromosome against significance, which is also not frequency. And that, my friends, is why you have to be very careful when you're looking at it. So this here is negative log 10 of p, which is significance. So scatter plots fundamentally are line graphs. So the same thing. They're more often drawn. If there's a line, you can connect them. If there isn't, you can do it. What you do is you draw a significance cutoff, and then anything about the cutoff you can say is significant. This is specifically okay. Great. Someone said scatter. I might have missed it. Sorry, Pranav. This is a Manhattan plot used for GWAS, one of my least favorite scientific methods. I think it's like really bad. It's one of those things that's really expensive and computationally intensive, and it's for all the people who don't know any biology to like be a biologist. I'm just kidding. That's not true. A lot of people are really talented. It's the best way to study human disease. But we'll talk about why it's bad in a later lecture. So this is a Manhattan plot from a GWAS study. Genome association study, for anyone who doesn't know. All right. Lastly, the thing they're going to ask you to do on the USABO is to draw conclusions, right? And normally when I draw conclusions, I like to draw it like this, right? I first begin with this like arc thing, and then I do a circle, and then how I'm feeling, I would do a round one, but you can also do a pointy one for the next thing. I'm just kidding. So when they ask you to, yes, I'm extremely humorous. Please, everybody, shower me with your compliments about my comedy genius. They ask you to draw a conclusion to figure out what does this data mean, right? And that's often challenging because they're gonna give you a bunch of results that all seem. Sorry, there was a gnat. I have houseplants and one of them has gnats, so now I have a gnat everywhere. It's terrible. What does this data mean? And it's really tough because they'll show you a graph where it's like, oh, when A goes up, B goes up. And you're like, oh, okay, then that obviously shows that A causes B to go up. Wrong. A does not cause B to go up. A is associated with B going up. That's all you can say. So I have created a 5-step check for conclusions. First, does the data actually say this, or is it an inference? Now, this is going to be tough because some questions ask you for which of these following can explain the data. And then in which case it's fine to be like A causes B to go up because that is a mechanism that explains like why it goes up. Right. All right. That's the thing about. But it's often if there's an inference. If I infer that A going up causes B to go up, then that's not a conclusion you can actually draw. 2, is the condition what is happening or something that could be happening? Sometimes they'll be like, which of these following explain situation? And the problem is they'll give you something that can explain it, but it's not actually what is happening. So that's kind of tough, right? What the hell does that mean, Kevin? What does it mean? So, like for example, one thing they'll ask is, if I add A and A leads to B going up, right? They'll say like, and like let's say that A is a metabolic poison. It'll be like, lowered energy results in higher production of B, and that does, but that doesn't actually. That's not actually true. The condition is A being present, not lowered energy, even if A is a metabolic poison. So again, you have to be careful thinking about, is that correct? Next, is the result what you think, or is the result something that leads to what you think? And this is similar to A, right? Like with, does A cause B, or does A cause something that causes B? This is more often an issue where I still want to point out the problem of, sometimes they will have a question where the answer is actually exactly what you think it is, right? But they won't express it that way. Instead of saying, A causes B to increase, they will say, the presence of a general thing which A is a part of causes B. Now, the first 2 rows kind of, you might think, okay, well, that's not necessarily the case. Yes, but you have to look at it in the context of all the things present. And again, these rows are going to be confusing. When we actually get to real questions, they'll be clearer, which Joseph will go over with you. And then 4, are there better choices? How does this one compare? And can you feasibly prove this one wrong? One of the things is, can this mechanism cause something else, right? You could be like, if you ever like this can, then that's probably wrong. It has to be it does. Like, mitochondrial bursting can cause energy to go down, and that can lead to B to go up. But that's not what you're looking for. You're not looking for like the distinct possibility of that. You're looking for a case where it does happen. So there's probably a better answer, although sometimes questions will have things like that. So you can't always be, and that question was bad, Claire. We're not talking about specific questions though. And then lastly, 5, is this biology correct as far as you know / makes logical sense? If it's not correct, if the thing is just wrong, then it's probably not. If you're like, this graph could be explained by ribosomes actually being made from lipids. Probably wrong. I would not pick that. Like, unless every other answer is like very obviously like a trap or is very obviously wrong, I wouldn't pick that one. I think again, I will say on some multiple selects, the strategy is eliminating all the ones you can, and the ones you don't know, just pick all of them. I would do that sometimes just because you never know. So we'll talk about strategy later, but these are some ways to check for conclusions, right? So it's important to know that the results are very limited often in what they're actually saying. All right, finally, for the last segment of today's course, we're going to talk about what is life, and this is something that we have to talk about because in case they ask the question, which the following is living or not, which has been asked once, I don't want you guys to get it wrong. So we're going to talk about this just in case, but it's very simple, and if you want to like space out, now's the time. Yes, this is Caspar David Friedrich, Wanderer Above the Sea of Fog. It's a very famous painting. If you guys took Euro, AP Euro loves this painting. This is an example of romanticism. And of course, if you took somebody on a date to a pointy rock with a lot of fog, they would love you because there will be no way for them to escape. So this is Wanderer Above a Sea of Fog. It's supposed to show like the infinite possibility of life and all these questions. But what I just see is some emo dude with too much time on his hands. So I want to open with a quote from my favorite scientist, E.O. Wilson. Those of you who were here last year, you saw this quote already. Humanity is exalted, not because we are so above other living creatures, but because knowing them well elevates the very concept of life. And this was written in his book Biophilia, which is not what you think it is. It's not what you think it is. It's talking about the inherent love for life that people have, right? Like most people, do not hate living things. There are exceptions. My mom notably hates all plants, all animals, and all microorganisms. I think that's a termite mound. I think it's a really big anthill. I don't think it's just sitting in front of like a mound of dirt. Although it does kind of look that way. E.O. Wilson actually is the reason why I went to Harvard. I read his book. I was like, this is like the smartest man I have ever met. And I spent 4 years there trying to meet him, and he's ancient. This man is so old that if you talk about like World War I, he's probably there. Actually, that's not that old, but he's very old, and so he's not around ever. He's a professor emeritus, and I was really hoping I could meet him. I spent 4 years trying to meet him. It did not work. His secretary is very protective, and she won't anyone meet him unless you're also famous. So that was that. I was not famous, and I still am not. So maybe one day. But he's called the Lord of the Ants. Really great scientist. He has a whole host of achievements, but whatever. So what's really cool about biology, and last year I had this whole thing about why biology is cool, but I didn't have it this year because I'm just gonna say, if you guys don't like biology, maybe don't take the course. I mean, it's a biology course. I don't know. But so I hope I don't have to convince you that biology is cool. But biology fundamentally has two principles to it: organization and evolution, right? First, but life is organized, right? Unlike your friends who are taking a physics or chemistry class, everything they're learning sucks. And now biology kind of sucks too, because at the end of the day, it is physics and chem. But because nothing is fun. They're like, oh, I have a chemical. If I add a little bit of chemical to another chemical, it makes a big chemical. Boring. Instead, what we have is a beautiful arrangement of things, right? And no offense to all the physics and chem people, but the organization of life, I think, is a truly, truly astounding element, right? Life is characterized by having lower entropy than its surroundings. Unfortunately, my room is pretty messy, so arguably it has higher entropy than its surroundings, but that's how it is. This organization is what lends so much ability for life to carry out reactions that are otherwise impossible. To the chemists and the physicists here, life, you can really reduce it down to a self-replicating chemical system. But I do think that's kind of a disappointing view of life. That's like viewing a cake as a bunch of carbs stacked on top of each other, held together by lipids. That sucks. Why would you choose to view it that way, right? You can choose to view life that way, or you can choose to view it as really cool. And that's what about evolution, that it betters itself over time. Now, I think for every people usually show the picture of the fish going up on land and becoming a guy walking, but I think this sort of wasn't better. This is a zucchini. Zucchinis are not meant to be this big. Zucchinis are stems, and this zucchini is so heavy it cannot stand up. It just lays on the ground, which is not good for the zucchini. But we as humans have driven natural selection to produce these giant zucchini. They're thinking, why don't we grow these? Why don't we eat this? Because it tastes bad. I've read about them. They're really bland, apparently. Like they're really watery, and so yeah, I guess if you like watery zucchini, I guess like go for it. But like, why do you like watery zucchini? What's wrong with you? I mean, I don't want to judge, but but like they can grow this big because we selected for it. We've gotten the genes to come together in this giant zucchini. Okay, someone says they like cucumbers, but they are not cucumbers, right? Fundamentally, zucchinis are different. I don't know. I really like zucchini. I have this great recipe for zucchini fritters if everyone wants it. Great recipe, super delicious, good for brunch. Oh, I have had some fire zucchini bread. Zucchini and dumplings? Isn't that like a ravioli then? Anyhow, excellent. So let's talk about it. Life is organized. And life is two ways, right? We talked a little bit about cellular organization and entropy. And we're going to talk more about that next week when we talk about chemistry. The chemistry lectures are very condensed, but they are also, as a result, kind of heavy. So be prepared for that, for those of you who don't have chemistry backgrounds. Hopefully it's not too bad. I'm cutting a lot of the really complicated chem stuff and then having Joseph cover it because he's my scapegoat for chem stuff. So what's more fun is the fact that life can be organized into this system of organization, right? We organize things on levels upon levels upon levels. And this allows us to create more and more complicated relationships, right? Not only are we organized atoms, those atoms are organized into specific molecules, of then made into organelles, and so on and so forth. Make sure you memorize this pyramid. I don't see why you wouldn't. It comes up like once or twice, so you may want to memorize it. And life begins to be a thing at the level of the cell. My bird was making some noise. I wanted to make sure it was okay. At the level of cell. So this is something that I think you like. I doubt anybody here like doesn't know, but if you have any questions about this, feel free to ask. What's more important, of course, is evolution, right? The gradual shift from one form to another, right? Obviously, in the Pokemon world, at some point, there was an intermediate between Gyarados and Magikarp, and then due to environmental factors, the intermediate was eliminated, thus preventing gene transfer. Now we have two species. I honestly kind of blame Pokemon for like a poor understanding of evolution. There's a very famous tweet that somebody was like, if mankind evolved from monkeys, how come I never seen a monkey at the zoo become a human? And I'm like, I don't think that's how it works. Whatever. It's not like Pokemon. Evolution is not. I think this picture is funny, but that's not how it works. So excuse me. So evolution has given us so much, right? Because I think organization is really cool, but evolution is what makes biology a science, and one that is very, very broad in its implications. As a result, it gives us this idea of unity in diversity. Despite life having so many different forms, right, we're able to see they are unified by this overarching theory. If there wasn't this, what we would end up having is we would just end up with a system where you have all these random things and everything we know about them is just random info. It's just trivia, right? The reason all your friends are interesting in their own right. But if you had a unifying theory for why they're your friends, that makes the things about them more interesting. At least I think so. This is also maybe the reason why sometimes my friends get weirded out. But I think it's interesting to know about things like that, right? And we get again some truly fantastic Cambrian fauna. We'll talk about them. Really, really cool. Obabinia. Oh my god, such a cool species. I think actually Herdia is not a great representative. They should have put like Cabroborastor. But anyhow, Peytoia and Atomalocaris, like famous species, and of course Tytopoda, the most famous of all the Cambrian fauna. Okay. Evolution fundament is this idea of descent with modification. Only the most fit survive, and as a result, the population gets better and better. Darwin did not say this quote, but it's humorous for those who are wondering. We'll talk about it in detail later. But what we're gonna do is I'm gonna talk about a lot of things from the context of fitness, right? We're gonna talk about right, like why things are, and in biology and philosophy are the only fields where you can ask the question why. You can't ask a physicist why does gravity exist. They can tell you how it exists, but there's no reason for it. But in biology, luckily, we can ask why, and we can ask this with the form of what are called ultimate questions, which is about evolutionary reasoning. It's kind of a mechanism still, it's how it came to be, but you can ask why about it as a result, and that's, I think, a really cool aspect of biology. I want to leave, oh my God. The Nats are really interested in joining the class, as you can tell, which is why you should join the class. The cell notification is one of those things that we're going to touch upon many times before we actually talk about it. So if you guys don't know what evolution is, maybe like send me a message, I can explain it. Okay. Oh, I don't know why that I did not put that animation in. I must have accidentally clicked the button for that. Okay, anyhow, finally, I want to leave you guys with this quote and a stock image. I couldn't find a non-watermarked version of this image, so whatever. It's from Theodosius Dobzhansky. He sounds like a Russian monk, but he's actually a very famous biochemist. And he said, nothing in biology makes sense except in the light of evolution. And then blah, 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 something else. All else is stamp collecting. And it's true. If you guys just kind of sat here and like accumulated information about biology, then like you would be like a book. And while books are great, we are people, and people are not books, usually. You can't flip them. If you try to open them up, they usually don't like that, and then they scream and they die. So what we want to do is we want to understand life from this context of evolution, and this gives us more power to take information and then create overarching systems, right? It underlies everything we do. There's evolution is what lets us say that the cells I study in my lab, which are not humans, have applications to human cells. Why? You can say a result we know from human disease can apply to animal or even plant disease. And I think that is what really creates a marvelous accumulation of information for us. So finally, I want to end on one last note. I will let people maybe go a little early, but I'm sure there will be questions at the end. I want to say one thing about this class. And, you know, I didn't know that this would be how it is when we started, but it kind of ended up being how it is. And I think one of the really valuable things about this class is the community that we've built around it. And, you know, it's a great place for everyone to study. This is our new logo. It was designed by Claire, who was actually in the chat this evening. You guys can explain why there's a weird border on it. I don't know. You guys give me this picture. You know, and I think it's this this culture of sort of collaborating and creating great works together and having a good time. I think there's no point in studying something if you don't like it and you're not having a good time. Because what's the point? You could do something else. You could study math with all the sweaty kids, right? And nothing is stopping you from doing that. And some of you probably will go through that anyway. But I hope that at least when you're studying biology, you know, you are having a good time. Yes, I'm just joking. The stereotype is that math kids are sweaty because they like focus really hard, so they sweat a lot. We've had a great community, right? People make fun of me. People make great art for this class. Somebody drew these hemoglobins. All the emotes on our Discord, a lot of them are drawn by students or made by students, right? We talk about, you know, nowhere else will you be able to find as good biology content as this extremely specific meme that Claire made about choosing to read Fox over reading Vanders, which are two of the most popular physiology books. We're not going to talk about this section, but we'll talk about the next section, right? This is a reference to in the past when last year when we asked people to turn on their cameras, nobody did. I know it's cut off and it says, teacher, turn on your camera, entire class. In terms of camera, we have no cameras. And then, you know, this is a joke because I like to mute people for spamming in the server, and not like I won't actually mute you. I'll mute you for like a minute. That's a good joke. So, you know, I think we have a great community, and I really hope, if nothing else, that you guys come join, come see what's up. talk to people, and get to know a little bit about what's happening. Again, Joseph will drop the Discord link again. And yeah, I hope to see many of you join. I think the kids are really excited to welcome you guys. They really want to do that for anyone who is planning on joining. So, Joseph, if you could put the Discord, or Claire or Pranav, really, you can do it too if you want. Yeah. One more thing. The back office doesn't know about this yet, so they're still here. They're gonna see this, but we've made merch for the class. Because last year we had a lot of great moments, we thought we would make some merch. You can buy the merch. It's got references to stuff about in session 1 of last year. So we've made some merch. It is based off the plastic bag design. You guys won't get the references. You will at some point, because I will make this joke again. This is a great joke. It's about James Watson, because he's terrible. But it's kind of cool. You can come buy it. We make like $0 off of it. It's just for fun. You don't feel pressured. It's only for fun. Yes. Basically, the joke is that James Watson is terrible and kind of a clown. We'll go over why in one of the genetics lectures. You know, buy this shirt if you like want to. It's kind of a fun thing. You know. So yeah, that is our course. I don't think that was a little short, but there used to be this like half the lecture was chemistry, so I cut all of that because I was like, I was just gonna cover it in the next lecture. Click the link that Claire sent at your own caution. I will not. So, homework for this week. Oh shoot, I should have said read chapters 2 through 4. We're not actually covering chapter 5 next week. Sorry about that. It's actually read chapters 2 through 4. Here, I will very quickly. I got the chapters wrong. Sorry. It's actually read chapters 2 through 4. Because we're not covering macromolecules. And that's because I have a biochemistry lecture now that talks about them. I'll talk about them from more biochemical perspective rather than just the chemistry perspective. We will be releasing a benchmark quiz for everyone to take, just the idea of how you are. Roy will go over the answers and then go over some like very general questions that anyone had from this lecture, talk a little bit about himself during his office hours on Tuesday, and maybe go over some like very basic review topics that I think maybe we want to talk about in a little more detail. And then in Friday's office hours, we're going to talk about problem solving. The benchmark quiz is not released yet. It'll be released, I said, like on Monday or Tuesday, which I know is not too much time, but we need time to like make the benchmark quiz. We have one, but we looked at it like last night and we weren't really happy with it, so we want to like edit it. So it'll come out like on Monday. It should only take like 5 to 10 minutes. It's very short. It's 10 questions, 10 multiple choice questions. So it's very, very fast. And then we'll go over the answers to it at the first Roy's office hours. And then again, Roy will go over some more basic things. He'll also go over some specifics of the U Toronto exam, which is an exam I recommend everybody takes at least for practice. And you can participate if they have it next year. And you know, normally all the Canada top prizes go to Americans, so everyone who is not a Canadian here, you have a pretty good shot. And then lastly, join the Discord. We'll put the link again. Come join. Email me. Let's have a conversation. I love chatting with students. God knows I like it too much. I've spent a lot of time staying after class chat with students. Yeah. Okay. Does anybody have any questions about logistics, about the class, anything at all? Thanks, Pranav. I am always online. Yes. The 11th edition has the same chapters 2 through 4 as the 10th, so you'll be fine. For anyone who has other editions, the homework for this week is just do it whenever. If you go to the Tuesday office hours, we will go over the answers to the benchmark quiz. So I guess if you don't have time to do it before then, we'll release it probably like tomorrow night. And there'll be an email about it, and we'll announce it on the Discord. There will be special roles. So once you get into the Discord for this week, there will be no special roles. But then starting next week, we will start having special roles for people who are formally enrolled in the class. You'll get access to a premium section of the Discord. The Discord is technically public. Anyone can join. I don't like share it everywhere. It's not meant to be like a public forum. But if you're not interested, you can still join. Come use my resources. There's a lot of free stuff posted there. So yeah, come hang out. We do picture of the day. If people remind me to, I'll post a picture every day of like diagrams and stuff I think are important or useful to learn. I'll try to keep it relevant to what we're talking about in class. What else is there to say? You have until next Saturday to pay. Also, if some of your friends didn't get a chance to sign up and they want to, feel free to encourage them to sign up. No, it's actually mostly not biology. There is very little biology on the server. I'm honestly very disappointing how little people talk about biology on my biology server. Yeah, we have a lot of hobby channels for like people who have hobbies. Like, there's a food channel. There's a art channel. The art channel is great. There's a nature channel. We're talking about nature, which is bio-y. Yeah, Claire, the coding channel is kind of dead. The coding kind of dead. Some of the channels. So we have the premium channels, right? And they're only accessible to people who have paid. That's still being set up right now. So we're working on that. Yeah. Claire loves DC. She convinced me to read Batman fan fiction. I regret it deeply, but it's actually really good. That's why I regret it. Come join. I talk about SCPs a lot for anyone who knows what that is. It's like this like online fiction thing because I'm a huge nerd. Someone asked, do I need to buy a textbook? You should ideally have a copy of the textbook. If every channel is locked, you have to react the welcome message to get in. So make sure you go to the welcome channel, react to the message, and then the bot will let you in. It's just to make sure you read the rules channel. Joseph can help you with that if you have an issue. Feel free to DM him or me. Yeah. Sometimes after class, I'll stay to chat with students about anything. Probably not today, simply because I have a bird show to go to tomorrow. I have a pet bird. You guys will probably meet him at some point. He's fairly curious. He's undergoing depression because I gave his brother away, because his brother was being not nice. So yeah, it's very sad. I kind of regret it now. I don't know how sad he would be. So I'm trying to comfort him. Look, it was it's a long story. You can come to Discord. I'll tell the story again. I have a really weird sense of humor. So for those of you who want to see more of that, come to Discord. I'll probably be on Discord tonight at some point to chat with people. Yeah, anyone have any other questions? Not about DC or Marvel, hopefully, because I've never seen any of those properties. I believe the best superhero movie is Birdman. That movie, right? Birdman? It's not. I thought it was a superhero movie. It is not. It is not a superhero movie. It's very much not that. Yeah, I thought it'd be about Harvey Birdman, like the guy on like the old cartoons who flew around. Yeah. Well, anyways, if people don't have questions, I will stop. I will stay until 8, but I'm going to stop the recording. So if people want to talk about other things, they can. Anyone have any other questions before I end the recording so that it gets in the recording? I'm going to count to 5. 1, 2, 3, 4, 5. All right. So, everybody, I hope you had a good time in the first class, and I look forward to hopefully seeing many of you return for the second.